Dara S. Manoach is a professor of psychology in the Department of Psychiatry at Harvard Medical School and MGH, and is a faculty associate at the Lurie Center for Autism. She's based at the Martinos Center for Biomedical Imaging, where she directs the Sleep, Cognition, and Neuropsychiatry lab. These webinars are made possible thanks to generous donor support. If you'd like to contribute, please visit our website at autism.org. And now I will turn this over to Dr. Manoach.
Thank you so much, Denise. I really appreciate it. I am very excited to share my research update on sleep with you today. And the title of my talk is Targeting Sleep Rhythms to Treat Neurodevelopmental Disorders. So, I'd like to start by defining a good night's sleep. Sleep is not a unitary state. A night of sleep can be divided into rapid eye movement or REM sleep in red and non-REM sleep, which can be further broken down into 3 stages. And during the night, we cycle through the different stages from lighter to deeper slow wave or non-REM 3 and 3 sleep every 90 minutes. And in the wee hours of the morning, sleep becomes lighter and we spend more time in REM sleep, which is characterized by dreaming, and also N2, stage 2 sleep, which is characterized by a specific brain rhythm called sleep spindles, which will be the focus of my talk. Now, my interest in sleep arose from my motivation to understand cognitive differences in neurodevelopmental disorders such as autism and schizophrenia. And the limitation to most studies of cognition is that they only measure they only measure cognition in cross-section at a single point of time. And while this provides a valid snapshot of function, it misses the critical aspects of learning and memory that happen offline, over time, and with sleep. After encoding, memories are not just stabilized in the brain like a snapshot, they're enhanced and transformed. And these consolidation processes happen offline, outside of conscious awareness, and can continue over days and even years. And much of this evolution of memories is mediated by sleep. Now, this is not a new idea. This is a quote from a Roman rhetorician of 75 AD. It is a curious fact of which the reason is not obvious, that the interval of a single night will greatly increase the strength of the memory, perhaps because the power of recollection undergoes a process of ripening and maturing during the night. But subjecting this to experimental study is relatively new. Starting with this landmark study of sleep, dreams, and memory in 2001 by my friend and colleague Bob Stickgold that made the cover of Science. In the past 20 years, there's been a virtual explosion of research in the role of sleep in memory. And this is Bob sleeping with his son Adam in the backseat of his car. And he's been a real pioneer in this work and has greatly contributed to sleep, our understanding of the role of sleep in memory. And I just want to give you a flavor of the breadth and importance of sleep-dependent memory consolidation to cognition across the lifespan. So sleep is very important for extracting the gist of materials learned at the expense of details. And I want to give you an example of that. So if I were to read you the words door, glass, pane, shade, house, open, frame, view, ledge, breeze, and curtain, there's a gist word that links that all together. And I'm just going to give you a moment to think about that. And it's window. And what sleep does is after a night of sleep, you tend to remember the gist words. And you forget, even though they were not actually on the list, and you forget the actual words. So what sleep is doing is it's resulting in a somewhat less accurate, but arguably more useful memory. It also prioritizes the recall of emotionally salient information. So in this example, if this were a parked car rather than a car wreck, it would be remembered much less well after a night of sleep. It enhances spatial navigation. So, you're given a task involving maze learning and you have a night of sleep compared to an equal interval of wake, you're going to do much better navigating after the night of sleep. It extracts insights and it facilitates language learning in infants. And together, this body of work has led to a paradigm shift. Not all of our learning occurs while we're awake. And sleep is an active period of cognitive function that facilitates learning and memory. And at this point, this idea has become so well established that Bob Stickhold's groundbreaking work has moved from the cover of Science to the cover of Scientific American. So when I first met Bob, he was studying the role of sleep in memory consolidation using a simple finger tapping task called the motor sequence task, which I'll abbreviate as MST. And this task is sort of like playing the piano. You put your hand on the keyboard and you type a sequence, in this case 41324, as quickly and accurately as possible for 30 seconds. You rest for 30 seconds and you do this 12 times. And if you play the piano, you've probably had the experience of hitting a wall with a difficult piece or a difficult passage, where you just can't get any better with further practice. And then when you return to it the next day, you find that you're a lot better. And this is exactly what happens with this task. So this is a study from Matt Walker. And what we're showing on the y-axis here is the number of correct sequences per each 30-second trial. Each trial is over here. And it doesn't matter if you train at 10 p.m. or at 10 a.m. You show a similar learning curve and you're sort of plateauing. So people are hitting a wall, they're plateauing, they can't get any better. But when they return to the lab the next day, they are significantly better. And this improvement depends on sleep, not just the passage of time. So if you train people at night at 10 p.m. and they come back 12 hours later after an interval of sleep, they're significantly better. And an additional 12 hours of wakefulness does not lead to further significant improvement. Similarly, if you train people in the morning at 10 a.m. and they come back after 12 hours after a day of wakefulness, improvement is not significant. Yet when you return after a night of sleep, you're showing significant gains. So I approached Bob about studying people with schizophrenia on this task because they have cognitive deficits that have not really responded to available treatments. And I wondered if sleep might be playing a role. So what we're showing here, again, number of correct sequences per 30 seconds. And I've put up a separate y-axis for patients with schizophrenia because overall they're typing more slowly. But what you can see is that they're showing a normal proportion and amount of learning during training. And this is consistent with the literature on other procedural motor tasks. However, when they come back after a night of sleep, only their healthy peers improve. They are not improving. They're not getting the benefit from sleep. So what I'm showing here is intact learning during training on the task and a specific failure of overnight improvement. This failure of overnight improvement occurred in the context of significantly reduced sleep spindles. And sleep spindles are a defining rhythm of stage 2 non-rapid eye movement sleep that are seen in the EEG on the scalp as 1 to 2 second bursts of activity in a waxing, waning envelope, hence the name sleep spindles. It sort of looks like a spindle. And sleep spindles are a key mechanism of synaptic plasticity that mediate memory consolidation during sleep. And importantly, in schizophrenia, the spindle deficit and the memory deficits were correlated, perhaps suggesting that reduced spindles are causing the reduced memory. I'd like to introduce the model that guides my research program. So my lab's work is primarily on the role of abnormal sleep rhythms in cognitive deficits in schizophrenia and autism. And I've started by talking about autism in this presentation because sleep rhythms have been studied longer and these links are better established. And as I hope to illustrate, it provides a useful framework for understanding sleep and autism. So what we're trying to do is to forge links, empirical links in causal chains leading from genes to circuits to physiology to symptoms, cognition to diagnosis. And we're hoping that this chain will lead to effective, mechanistically guided treatments. So my sleep research program started with the finding that, unlike their healthy peers, people with schizophrenia fail to improve their performance on a simple typing task after a night of sleep. So this demonstrates a deficit in sleep-dependent memory consolidation in schizophrenia, and we linked it to reduced sleep spindle activity. Importantly, the spindle and memory deficits were correlated, which, together with evidence from intervention studies, suggests that they are causally linked. Sleep spindles are generated in a deep structure of the brain called the thalamus, and they are propagated to the cortex via thalamocortical loops. And the spindle deficit in schizophrenia suggests a problem with this circuitry. So I just want to say a little bit about the circuitry. So sleep spindles are generated in the reticular nucleus of the thalamus, which is part of the thalamus that is strategically localized between the rest of the thalamus and the cortex. And so this strategic localization allows it to guide thalamocortical communication. And during sleep, it generates sleep spindles, which are propagated to the cortex via these thalamocortical neurons. During wake, information from the senses is relayed from the thalamus to the cortex. And like a net, the TRN gates this flow of sensory information, only allowing through relevant information. So for example, hopefully it's allowing in what I'm telling you today and stopping the relay of information that's irrelevant, like noise from the ventilation system, or in my case, noise from outside my window where they're apparently working on something in my backyard. Pathology studies show that the TRN is abnormal in schizophrenia, showing both a reduction of neurons and also perineuronal nets, which are the scaffolding that nourish and protect these neurons. So what might we expect if, as a result of a broken TRN, gating was disrupted in schizophrenia? One possibility is that there'd be increased and less filtered relay of sensory information from the thalamus to the cortex, leading to a fragmentation of attention, which is a hallmark feature of schizophrenia and is captured by this patient's self-report. Recently, I've noticed that noises all seem to be louder than they were before. I notice it most with background noises that are always around, but you just don't notice them. Now they seem to be just as loud and sometimes louder than the main noises that are going on. It's a bit alarming at times because it makes it difficult for you to keep your mind on something when there's so much going on that you can't help listening to. It may also lead to impaired sensory gating as measured with EEG. Normally, there's an attenuated brain response to the second redundant stimulation that occurs close in time. And people with schizophrenia and their relatives have reduced gating of this redundant information. So here's sound one, here's sound two, and the gating response, the brain response, the event-related potential looks equal. Impaired gating may also account for the well-replicated finding of increased communication between the thalamus and sensory motor cortex in schizophrenia. So they're showing increased communication, and this is measured by resting state functional connectivity, MRI, and the same sample also had reduced spindle density. And what I'm just showing you is a topography with the EEG montage. So each of these circles is an electrode, each of these labels is an electrode, and the circled ones were significantly lower spindle rates in schizophrenia. And importantly, the spindle deficit and this hypercommunication between the thalamus and the cortex were correlated, consistent with the hypothesis that both decreased spindles and increased connectivity reflect the same thalamocortical circuit dysfunction. So this hypercommunication on functional connectivity MRI gives more evidence of a thalamocortical circuit dysfunction in schizophrenia and links it to spindle deficits. So theoretically, we have a mechanism, thalamocortical circuit dysfunction, that may contribute to several important features of schizophrenia: spindle deficits, reduced sensory gating, and thalamocortical hyperconnectivity, as well as reduced sleep-dependent memory consolidation. In schizophrenia, genome-wide association studies have identified CACNA1I as a risk gene for schizophrenia. It encodes the CAV3.3 calcium channel, a channel that's found only in the brain and particularly in the TRN, the thalamic reticular nucleus, which again generates spindles. And disabling CACNA1I in mice results in a spindle deficit. And of course, CACNA1I is only one of many genes that are involved in spindle deficits. And colleagues at the Broad Institute, led by Jen Pan, have created mice with a mutation of this gene, the same mutation that's seen in schizophrenia. And what it does is it leads to a reduction of the expression of the CAV3.3 calcium channel on the cell membranes of neurons in the TRN, reduced burst firing in the TRN, which is the firing that leads to the generation of spindles, and a corresponding reduction in spindle activity. So WT is the wild type animals, which don't have the mutation, and these are the knockouts, and you can see the reduction in spindle rate or spindle density, number of spindles per minute. And this team is now working to identify drugs that correct the function of this channel with the goal of increasing spindles and improving memory in schizophrenia. Risk genes for autism are also implicated in TRN dysfunction. For example, PATCH-G1, which is mutated in about 1% of individuals with autism, is selectively expressed in the TRN early in postnatal development. Recording from the TRN, directly from the TRN in mice with PATCH-G1 deletions shows that they have reduced burst firing, Again, wild type and knockout. Wild type is in black, knockout is red, and a corresponding reduction in spindle rate. These mice also show reduced TRM gating of visual information from the lateral geniculate nucleus, the visual nucleus of the thalamus. So they have, more flow of this visual information to the visual cortex and a corresponding increased vulnerability to distraction on a visual attention test, thereby directly linking gating problems to attention deficits. Importantly, both the spindle and attention deficits can be rescued by a compound that corrects channel functioning, raising the possibility of effective treatment. So this raises the question, how might a genetically mediated hit to the TRN early in development predispose to a neurodevelopmental disorder like autism? Rodent studies are providing clues. During gestation, axons that connect the thalamus to the cortex all have to pass through the TRN in red, which guides them to their terminations in the cortex. And as early as the first postnatal week, spindle bursts, which are generated in the TRN and are a precursor of adult spindles, establish and refine this thalamocortical circuitry. So in this way, early TRN dysfunction may affect circuit development and set the stage for autism. So motivated by findings that ASD risk genes affect the TRN, we started looking at sleep in autism. Up to 80% of children with autism have sleep disturbances, which exacerbate symptoms and lead to worse daytime functioning. And Dimitris Mylonis in the EEG captain lab has reanalyzed data from an already collected small home sleep EEG study of 9 to 17-year-olds with autism spectrum disorder. So on average, both groups showed over 8 hours of sleep. But people with autism had worse sleep quality, characterized by reduced sleep efficiency, which is just a ratio of how much time you're in sleep versus how much time you're in bed here. And they showed much more time in bed to get the similar amount of sleep. They showed increased WASO or wake time after sleep onset, so suggesting they have increased arousals, and increased latency to onset sleep, as well as a higher percentage of time in non-REM stage 1 sleep, which is the lightest sleep stage. And I am just going to have to close the window because my TRN is not efficiently gating out the noises from my backyard. They're now playing music. Just give me 1 second. The window's right here. Forgive me. All right. So we also found other evidence of thalamocortical dysfunction in autism. They showed reduced sleep spindle rate in stage 3, non-rapid eye movement sleep. So this is TD is in blue, autism is in red, and these are frontal versus central electrodes. We also found reduced coordination between the thalamic spindles and cortical slow waves. So what I'm showing you is a canonical slow wave, and what's happening here is that in autism, the spindles are peaking later in the slow wave upstate, and they are more variable in their in the time of peaking. This is consistent with the disruption in the dialogue between the thalamus and cortex during sleep. During wake, similar to what we see in people with schizophrenia. Functional connectivity MRI studies show increased communication between the thalamus and sensory cortex, including, so here's somatosensory cortex, including auditory areas. And we've a finding which we've replicated. And our hypothesis is that there may be less gating of auditory information to the cortex as a result of TRN deficits. In ASD, less filtered relay of sensory information, reduced gating from the thalamus to the cortex may lead to sensory overload and to sensory sensitivities. And using EEG, we show that compared with their typically developing peers in autism, gating is much more variable. So on the x-axis is the ratio of the response to the second compared to the first sound presented during this sensory gating task. One is an equal response, and less than one is an attenuated response to the second redundant stimulus, which is what you would normally expect. And some of our ASD participants are actually sensitizing to the second stimulus instead of attenuating their response. But obviously, this is a very small sample, and we need to see whether these findings hold up in larger samples. What about sleep-dependent memory consolidation in autism? So in either of the studies in our lab, this was a spatial memory task or the MST, there were no deficits in sleep-dependent memory consolidation. And this is also the case in the literature. While typically developing people showed the expected relations of sleep spindles with memory in both tasks, people with ASD, if anything, showed the opposite relations. So this left us scratching our head. If not memory, what is the function of spindles in autism? And there is evidence that the TRN gates sensory information during sleep as well as wake by generating spindles to prevent awakenings. And this may be what allows you to sleep through a garbage truck outside your window, but makes you sit bolt upright in bed if your infant utters a peep, this sort of sensory gating for what's relevant and what isn't. So this inspired us to examine the relations of spindles to sleep quality in ASD. So in ASD, fewer spindles predicted worse sleep quality as measured by WASO, wake time after sleep onset, and also the percentage of time spent in stage 1 sleep, which is the lightest sleep stage. And this was true in autism, but not in typically developing children. And what this suggests that is in autism, spindles may be acting to preserve sleep, and that spindle deficits may contribute to sleep disruption. So these findings motivate our current work. We are studying the links of sleep disruption in children with autism to spindle deficits, circadian disruption. We are also examining thalamocortical circuit function during the day by looking at sensory gating abnormalities using EEG and their relations to sensory sensitivities. We are also collecting pilot data for a possible clinical trial of the effects of melatonin on sleep physiology. Despite its widespread use, there have been very limited studies of the effects of melatonin on sleep physiology, plenty on sleep quality. And our clinical trial rationale is evidence that melatonin release is reduced in autism, and that in typically developing people, melatonin can improve sleep quality and increase spindle activity and realign circadian rhythms. To overcome the curse of small sample sizes, we are scaling up by using wearable sleep headbands and actigraphy devices, which measure movement that can be worn at home. Because lab-based sleep studies are burdensome and expensive, we've been funded by the Simons Foundation to test wearable EEG devices that can characterize spindles at home against the gold standard polysomnography. So what we did was we had adolescents, typically developing adolescents, visit our lab for nap studies in which they wore both the sleep headband and had simultaneous polysomnography or sleep EEG. And all the commercially available devices we tested had good agreement, with sleep staging. And all of them could accurately detect slow waves. Here's the downstate, here's the upstate, and the spindles riding on top of these slow waves, this spindle activity, which is coupled with slow waves. Some of them can also deliver precisely timed sounds, auditory stimulation, which is a potential treatment for spindle slow wave coordination deficits. Brian Baxter in my lab is testing closed loop auditory stimulation during sleep. So closed loop because it's timed precisely to the upstate of slow waves. So what people do is they come into the lab for a nap. They first are tested with the MST, trained on the MST. They then have their nap monitored by polysomnography. And then they're tested to look at sleep-dependent improvement. And during the nap, we detect in real time the downstate of the slow wave, and then we time stimulation using a prediction algorithm to the upstate of the slow oscillation, the slow wave. So what we're doing is we're playing a brief burst of pink noise timed to occur in slow wave upstates. And what we found is that CLASS, I'll abbreviate as CLASS, closed loop auditory stimulation during sleep, has very strong effects on sleep rhythms. So in black is our sham condition. We're only detecting slow oscillation upstates, but we're not stimulating them. And here's the detection point, and here's the stimulation point. And what we can see is that in red, the upstate stimulation is augmenting the ongoing slow oscillation. It's leading to a downstate and then another slow oscillation upstate. It is also increasing sigma activity, which corresponds to spindles. So we can see that it is increasing sigma activity over sham in the same time as the upstate of the slow oscillation. What about memory? So CLASS increases spindle slow wave coupling. And this is again the EEG montage. Each black dot is an electrode. These green dots are where coupling density or the rate of slow wave coupled spindles is increased. This increase in coupled spindle slow wave density correlates with MST improvement. And this was true in all conditions. So all conditions showed similar improvement on the MST after the nap. So this CLASS did not lead to better memory, unfortunately, despite improving coupling. And we're now trying to learn from this data to optimize CLASS. And it turns out that timing matters. So the closer the stimulation occurred to the upstate, the more likely it was to evoke a slow wave. And the same was true for spindles. The closer it was to the upstate, the more likely it was to evoke a spindle. So timing matters, and we're trying to more precisely targeting the stimulation based on individual physiology, so a precision medicine approach. And we're now also piloting the use of headbands to deliver auditory stimulation overnight at home. And this will also allow us to stimulate for a whole night rather than a nap. It just could be maybe that in a nap, we just don't get enough stimulation in to increase memory. So we are also using our headbands to scale up our study of sleep and autism and to reach communities across the United States. And our goal is to get all 50 states. So what we're doing is we're shipping the headbands by Federal Express and providing phone and video support to use them. So we're collecting a total of 3 nights each in typically developing children ages 12 through 19 and people with autism, same age. I just want to show you some preliminary data from that study. As a whole, we're not seeing a spindle deficit in either stage 2 or stage 3 sleep. However, when we break our sample into males versus females, what we're seeing is that there is a deficit, but it's constrained to males. And we wonder if some of the inconsistent results in the literature may arise from the proportion, may depend on how many females were included, if they're sort of acting counter to one another, because if anything, in females, it looks like they're showing greater spindle density. And this is very preliminary, but I think what this is illustrating is the importance of having a large sample so we can begin to understand the factors that determine variability in these sleep parameters. You know, so for example, in our children's hospital, it was all male. The children's hospital home sleep study was all male participants, and we did see a spindle deficit. These sleep headbands and the large samples may also tell us who is most likely to benefit from interventions aimed at improving sleep rhythms. We have also adapted our protocols for people with profound autism and genetic syndromes that are associated with autism. What we do is we're doing consent electronically over Zoom. We are then bringing people in for a characterization visit and we send families home with a mock headband, which is just a headband that has the hardware removed so people can practice with it and family and the child can get used to wearing the headband overnight. They can be desensitized to it, and this desensitization process can take 1 to 2 weeks. We then have an ERP visit where we measure sensory gating and we send families home with the real headband, and we record 3 nights of sleep at home with the headband, and then people return the headband by mail. And if necessary, we can skip the ERPs and do this study completely remotely. So we are turning our desensitization protocol into an app to make it more usable by families. And the app has the desensitization protocol starts, with small steps of touching the headband, having a family member wear the headband, wearing the headband itself, and lying down with the headband, and finally sleeping with the headband. This was designed by Nicole Simon, and we try to individualize the protocol based on the child involved. So what I'm trying to illustrate is that we're developing tools to make sleep studies more accessible to understudied populations who may not be able to come to the lab by virtue of location or level of function. And it was surprising to me when I looked through the literature, trying to find studies of sleep physiology in people with profound autism and those with genetic conditions related to autism, what a paucity of sleep physiology studies they were, despite studies showing that there's tremendous sleep disruption as measured by questionnaires. So, in summary, we will be studying people with genetic mutations linked to autism, specifically Phelan-McDermott syndrome, Fragile X, and 16p11 deletion syndrome. And in collaboration with Shantanu Jadhav from Brandeis University, we're going to study rats with mutations of these same genes. And if there's a good correspondence, this would be very useful because it would allow preclinical studies that would allow rats to be used to evaluate treatments before moving to clinical trials in humans, such as class or drugs or compounds or supplements such as melatonin. And the goals of these treatments are to correct thalamocortical circuit dysfunction, increase spindles or improve their coordination with slow oscillations, improve sensory gating, which I think will be linked to an improvement in sensory sensitivities, and restore sleep. And the goal is to try to improve the lives of people with autism as well as their caregivers. And I just want to acknowledge the team at the Lurie Center who is running these studies. So there's Demetrius in my lab, Annabella, and Nicole, who are both research coordinators. Paige Hickey is our postdoc, and Newmeyer is a neurologist at the Lurie Center, and Nicole Simon is a behavioralist who took the lead in helping us to develop the sleep headbands. Also want to acknowledge my funders, particularly the Simons Foundation Autism Research Initiative, which funded our headband and cross-species studies. And finally, I'd like to thank our participants and their caregivers for their participation. And finally, I would like to just end with a sort of ad saying we are actively recruiting study participants. And I think Denise may have put the link in the chat, and there is the QR code that will link you to find out more information on the research we're doing in general, and also the studies where we're recruiting volunteers. So I thank you very much for your attention. Please forgive my voice, which is, I'm getting over a cold, and the break to close the window to prevent noise. And I'm very eager to hear your thoughts and questions. Thanks so much.
All right, great. Well, let's keep that slide up. I know people are asking for that, and I will post it again in the chat, and we'll be sure to send it out with our link that we'll send out tomorrow. We will send the playback link, and we'll include that link. So just a couple of things. The first question, it wasn't really a question, it was just a comment. Attendee was saying autism is not the same disorder as schizophrenia. It's important to separate these two conditions. Just because someone is autistic does not make them schizophrenic. So I know you talked about that a little bit. Does this come up in research conversations? Do you have any thoughts on that?
Yeah. So, my first take on this was to try to impose the template from schizophrenia onto autism. And that thoroughly did not work. Because people with autism, they're both neurodevelopmental disorders. Schizophrenia has a later age of onset, but has all sorts of prodromal features that are apparent earlier in development. But they look very different. So coupling in spindle slow oscillation or slow wave coupling in schizophrenia looks pretty normal. Yet in autism, it really does not. Spindle deficits are much more pronounced in schizophrenia than autism. And I think the contrast is really instructive to the extent that it helps us understand the mechanisms because these different endophenotypes, these sort of hidden phenotypes that you need EEG or other techniques to measure really imply, despite the genetic overlap, and there is some genetic overlap, implicate different mechanisms. And I just want to sort of sit down and really have time to think about all these differences and what they're trying to tell us about different mechanisms. So I really appreciate that comment.
So I will start at the top here. We have a lot of questions. So this first question, are sleep studies beneficial for children who have very early waking? Even if bedtime is being pushed back, our child still arises between 3 and 4 every morning.
So I'm not a clinician. I'm not doing sleep medicine. I am a neuroscientist. Actually, I'm a clinician. I'm a neuropsychologist, but I'm not wearing that out right now. But I'm not a sleep medicine clinician. And so, yeah, I mean, I don't know if a sleep study would be beneficial in that circumstance, to be very honest. What we're looking at is not sleep quality per se, but the brain activity that underlies sleep. And we're trying to understand what may be associated, what physiology may be causing a disruption in sleep. There's also circadian rhythm disorders in people with autism that I haven't really addressed in my talk. And one of the things that I hope that future research will bridge is the link between sleep physiology, sleep science, and circadian science, because these two are intimately related, and there's very little research that tries to bridge them and understand how they interact. So I think the questioner has pinpointed a very important lack paucity in the literature. And I'm sorry I don't have any more useful advice to give in this situation.
Yes, and this is a question that came up a lot in the Q&A. So I think if you were a parent of any age, so whether it's a young child or somebody who's in their 20s or 50s, what type of expert should they go see? Like, would they go see a neurologist? If possible, a sleep specialist in their area, but a medical doctor is what we would typically refer to, correct?
I think a pediatric neurologist would be the way to go.
For a younger child, yeah, and then an adult, and then for an adult and a general neurologist.
Yeah. General neurologist, or we also have in psychiatry people who specialize in sleep. So I think you just want to find the person who has the most knowledge of sleep and autism.
So trying to reach out maybe in your community and find out who people are going to for that.
Yeah, I would, as always, start with the PCP for these sorts of referrals. I think we have excellent clinicians at the Lurie Center who might be able to direct people appropriately. Again, I'm not doing clinical work in autism, so I understand. I'm hoping that future research will address some of these needs, but I don't have the answers to some of these clinical questions, sadly.
So this is a question about elderly adults, and they're asking about, have you seen any current research on seniors on the autism spectrum who demonstrate some of these differences? Have you seen anything in that age group?
So we've not looked at elderly adults. Our typical studies have a cutoff at age 45. So I've only presented data primarily from children in this talk. And that's because sleep spindles change over the lifespan, and the dependence of memory also changes over the lifespan. And I think sadly, there's not a lot of research on, autism and overdevelopment, especially for elderly folks. And I know that was a topic of a Lurie Center retreat this past Friday, was the dearth of studies, research on elderly people with autism.
Yeah, and there was actually last fall in Canada, in Vancouver, British Columbia, they had a think tank where they really focused not just on sleep, but all sorts of co-occurring medical conditions that seniors are experiencing, and looked into that. How do we improve the quality of care? And I know some of those people there was crossover between those two groups. I would also say to this person, she had more specific questions about her specific experience that she's having, and looking for a neurologist in her area or a psychiatrist who does work with that population may be a little different approach. It sounds like she's really been directed that she has to be in a CPAP and these different approaches, but maybe there would be a more holistic approach if she tried a different type of specialist. This next question.
I'll just add that, sleep changes normally, with aging. So in typically developing people, sleep changes with aging, and there's less slow wave sleep, and I don't know the degree to which autism veers from that trajectory, because that research does not exist.
Yeah. All right, so this is referring to some of your data. This person says, we can see in the diagrams that GABA has a function in sleep. Is there any evidence or research that you've seen about gabapentin or melatonin or these other supplements that people use to try to address sleep deficits? Have you seen research about that? Are you aware of any resources that they might go and look at?
So we have actually done trials of non-benzodiazepine sedative hypnotics that act on GABA A receptors that are expressed in the thalamus. And we've done them in healthy people and in people with schizophrenia. And what we found was that although, this is azopiclone or Lunesta, the butterfly drug, although it increased sleep spindles, it disrupted the coordination of spindles with slow waves. And so it did not wind up improving memory. So I think I am a little bit skeptical that there's a free lunch out there in terms of sleep meds. Melatonin, which is a natural supplement, there is evidence for increase in sleep spindles. I don't know that there's any evidence for increase in coupling of spindles with slow oscillations or effects on memory. I think what I'm most excited about is our closed loop auditory stimulation studies, because we're not ready to do them in people with autism yet. But we will be doing them in rodent models. But that is sort of a more, it's taking advantage of a more endogenous mechanism by just presenting soft sounds. So yeah, so I guess that's what I'm most hopeful about, is things that because I think some of these sleep medications that affect GABA may have unknown or unexplored side effects on memory and the coordination, this really delicate orchestration of sleep rhythms, both spindles and slow waves, and also hippocampal ripples, which I didn't have a chance to talk about today. So I think I'm a little bit skeptical.
Okay. This next person, I'm just going to read this because she's not asking for advice, just making a comment. They're saying, I have hypersensitive hearing. However, after 69 years, I have been able to filter out a lot of noises I hear to be non-eventful. I've tried a completely dark and quiet sleep environment, but my sleep is even worse. I need constant white noise in the background to have any beneficial sleep at all. I wore, I'm not going to say this right, actigraphy watch back in 2008, and I was told I have no circadian rhythm. So just a comment from somebody that's their experience that they're having.
That's exactly why we're studying it. I mean, it sounds so much like this gating or filtering problem that we're trying to get our heads around. And I wish we were further along with this in we did have better answers, but that sounds like also the intersection of circadian issues as well as sleep issues in terms of filtering incoming sensory information to screen out and protect sleep from disruptions by irrelevant information.
All right, lots of questions about the study. I'm clearly getting to the section where you were talking about it. One question they had, and I apologize if I missed it, you may have addressed this. If people come in and participate in the study, do they get some information about the findings from their particular child's experience, or is that data just being put together and then reported generally?
So we will, we can't give clinically relevant information. But what we do give is like sort of a picture of an individual sleep spindle. We share an article on that I wrote with Bob Stickold, geared to children, on the role of sleep and memory. And we will include and inform people of any publications that come from the research. But in terms of giving individual feedback on sleep, I think we can also give how much time was spent in each sleep stage. I think we may also do that. But nothing particularly clinically actionable.
Okay. So another comment here, this is a person who's saying that they're looking for adult research and they're available and really want to participate when there is some. So just a more reinforcement for that if you have any colleagues looking for that. This is just a question about, you can talk a little bit about the instruments you're using to assess autism for validation. So how are you identifying, for example, profound autism for your study? What instrument are you using?
So we're leaving it to clinicians to do that. I think we're basing it on IQ, the presence of challenging behaviors, and whether the child is verbal or not. So we're using the same definition as the Autism Science Foundation for profound autism. I think that's a very difficult clinical judgment to make. And in reality, we're including people sort of across various levels of function, and we're not sort of categorizing except for our more remote study where we're limiting it to idiopathic autism without significant intellectual disability, just because I think it's harder for us to work with families remotely than if we actually see them in person.
So this person just is looking for clarification, wants to make sure they understand correctly. Sleep spindles in girls with autism are not affected like boys with autism, correct? So the sleep spindles would not be the culprit for sleep disturbance for girls with autism.
So I presented this data with caution because it's very preliminary. Yep. It's the first data of its kind. So that would be a reasonable conclusion to draw from this data, but I don't think we're there yet. And I think girls are different in ways that we don't yet understand. Okay. And tend to be under-identified with ADAS, but are not as under-identified filling out questionnaires or by their own reports. So there's a real discrepancy there.
Okay. This is a question, and I know we're almost out of time, but I'm going to squeeze in a couple more. Have you seen a study done related to your work that also addresses the gut-brain axis and sleep? I'm sorry if I missed it. You may have mentioned it. This is a person who joined late. So.
No, I'm sorry. I have not seen any studies that have addressed sleep physiology as opposed to sleep quality in relation to the gut-brain axis. So sleep physiology, I'm just talking about the brain activity underlying specific sleep stages as measured by EEG. I've not seen any studies trying to understand the the gut-brain interaction with regard to sleep on sleep physiology.
Okay, this is just a statement, this person saying after years of sleep deprivation, their child started sleeping soundly after improving their diet. So it sounds like they had help from a nutritionist, and they're part of a study now that's at the NIH that's looking deeper into that. So that's interesting. And then finally, this last question, or just a statement, my daughter was oversleeping, so 14 plus hours a night. Overnight sleep studies showed no REM sleep occurring. They hypothesized this was due to medication that this person was on, risperidone and SSRIs. Just pointing that out, that's another interesting thing to be looking at and thinking about.
Yeah, it's a huge compound in studies. And again, in our headband studies, you know, we're trying to be as inclusive as possible. We're hoping to have enough participants that we can begin to understand what some of the factors are that lead to different responses or different profiles of sleep.